Today's video will be a slightly harder topic regarding physiology, which is about neurons and how the specialized cells transmit signals throughout the body. The objective of this video is to cover how to draw and label one, a basic neuron, talking about how an action potential works and how it travels along an axon, briefly talking about myelination, and focusing on how synapses work, with relevance to how chemical synapses are responsible for muscle contraction, and talking about how acetylcholine, a neurotransmitter, allows for this to happen. This would nicely lead on to an in-depth detail of how serrated muscle contracts once signaled. First off, we'll learn how to draw a very basic neuron. This depiction here of a motor neuron is what the lesson will be based off of. So we'll start off with a basic neuron, which consists of its nucleus, a very long, unusually shaped cell membrane, which allows for it to carry out its specialized role. There we go. In terms of labeling, myelins we'll be discussing about later, but in terms of labeling, we'll just label the nucleus, which is responsible for cellular control, the large head, known as the cell body, which is where the vast majority of the cytoplasm is concentrated. It is also known as the soma. Dendrites, which receives signals, either chemical or electric signals from other neurons that comes before it. The axon terminal, where synapses occur between the receiving cells and or other neurons, and the thing that makes this special cell stand out is its axon. And regarding to how action potentials are generated, it is generated at this region here at the base of the soma, known as the axon hillock, where the membrane potential, where the action potential first builds up, is generated through a graded potential being received from the dendrites and passing it along the axon. And action potential travels along the axon. Moving on to talking more on action potentials. To begin with, action potentials are generated depending on the strength of stimulus. The stimulus would manipulate graded potential, which allows for an action potential to be fired off. These stimulus can vary depending on where the neuron is located, the kinds of stimulus, and what the main function of the neuron is, depending on whether it's connected to a muscle cell or supposedly neighboring other neurons, or sending signals along the spinal cord. But we are looking specifically at a motor neuron and its relation with a muscle fiber. During the first phase, of which is known as a resting potential, where the potential within the membrane varies between -70 millivolts and -55 millivolts. The factor that is responsible for there being a potential in the first place is due to the presence of sodium ions and potassium ions. And to remember where they're located, sodium ions are located outside the cell membrane, outside the axon, and potassium are located inside. Sodium is slightly more positive, so outside is positive and the inside is negative. The sodium ions and potassium ions are being transported in and out the cell during this phase, through a process known as active transport, where with the use of an integrated protein, as shown in the first picture here with this weird protein. What is powering this this mode of active transport is the breakdown of the hydrolysis of ATP to ADP. As a protein, as the membrane protein itself has a kinase group that allows for this to happen. The inside of the cell is slightly less positive than which allows for the inside of the cell to be slightly less positive than the outside, as more sodium ions are exiting. That's why there's sodium on the outside. More sodium ions are exiting the cell, and less potassium ions are entering the cell. So there's more. The reason as why the membrane potential fluctuates between minus 70 millivolts and minus 55 millivolts is due to a not strong enough stimulus that has not yet surpassed a threshold of minus 55 millivolts. Specifically within a motor neuron that is connected to a muscle fiber. The protein itself is a special ligand-gated protein, which means that it undergoes a change where the protein opens up once a neurotransmitter binds to the active site of the protein. Here's a neurotransmitter. And our positive ions can go in. Once there's a higher concentration of stimulus, there's going to be more and more sodium ions that would enter the cell, which would allow for an increase in the membrane potential. And once the membrane potential reaches a threshold of minus 55 millivolts, known as the excitatory threshold, it enters a second phase of depolarization, where there are voltage-gated sodium ion channels that allow for even more sodium ions to enter, as depicted in the second image here, where this yellow protein channel is opened in response to a membrane potential that is over negative 55 millivolts. This again is known as the threshold of excitation. As more and more sodium ions rush in, the membrane potential reaches a peak action potential over here, which is more or less 30 millivolts. Once the peak action potential is reached, there is a third phase that comes after the excitation. Third phase that comes after the excitation pure threshold, known as repolarization, which is the third image, depicts here, where the sodium voltage gated channel will take an inactive form, as in response to a higher membrane potential reached. This would this would briefly close sodium channels in order for this would briefly close sodium ion channels, and no sodium ion channels will enter, but voltage gated potassium ion channels, as depicted by the orange looking integral protein here, will open up to allow potassium to be released, which repolarizes the membrane and brings it back down to a level of restoration, to allow the membrane potential to go back to its original charge. The final phase is where the region of the cytoplasm restores itself, which as can be seen in the graph here, is in a fixed phase of repolarization. It repolarizes itself back to normal and cannot be stimulated by anything during this very short period of time. So no stimulus can either open or close any of the gated ions, which would allow for the repolarization of the region of the axon back to a normal phase of the resting potential. How the action potential travels through the axons once generated is by a chain reaction of this event. As this is measured at one region, there are other regions along the axon that also experiences similar action potential effects. This continues to happen as you go down along the axon till the axon terminal, where the action potential ends up either releasing a chemical or an electrical synapses.